get a good feeling, yeah.
I believe the place to start talking about human physiology is with talking about the functions of membrane proteins. Now, you remember, when we started talking about anatomy in the fall, that if you're going to talk about how you build a human body, you got to talk about 4 organic macromolecules, right? Carbohydrates, lipids, proteins, and nucleic acids. So each of those plays, their own role, and we talked about those from different perspectives during the semester. But I made a point during that opening discussion of anatomy to tell you that if you're going to talk about, ultimately, if you're going to talk about how something in the body is built or how it works, you're going to have to talk about the proteins. For example, if you're going to talk about the structure of bone or any connective tissue, how something is constructed, why your ears look like they do, what the connections are between bones, how things are held together in your body. You're going to talk about collagen as a protein. We do that as structural protein. If you're going to take a drug, for example, let's say you're going to take a drug because you've got some inflammation. They're going to give you a corticosteroid. They take a little cortisol, get the shot you need because it's swollen or something. Are you going to take cortisone by mouth, some prednisone because of whatever infection you've got or whatever to reduce inflammation? They're not going to let you take that very long because that drug, that glucocorticoid drug that you're taking, that prednisone and cortisol, is going to reorder your physiology in such a way that your body builds sugar over against building protein. It's a process that we'll learn later in the semester called gluconeogenesis. But the reason I bring it up here is because the reason they're not going to let you take that drug long is because it's going to interrupt your body's process of building proteins. Your bones will get weaker. Your muscles get weaker. Things don't work in your liver. And on the list goes whenever you start stealing the carbons from the protein factory and using them to build sugars with. That's what the glucocorticoids do. And so it's a good way of thinking about this. If you're going to talk about how a body's built or how a body works, you've got to talk about the proteins. Now this picture shows the freshman biology image. I think this one was actually in the mirrored text of a cell membrane. And you can see all the proteins, these purple things present in the cell membrane. Now, as we looked at these proteins in anatomy, I said, this is the reason why they call the cell membrane a fluid mosaic model. A mosaic is a piece of art made up of a lot of different kinds of pieces. Like a mosaic would be built out of glass shards put in order so that you make a picture out of them. If it's fluid, it means it's moving. And so that's what's going on here in the cell membrane. These proteins in the cell membrane are moving about, and they do all kinds of things. This class will talk about almost all of them between now and May. The things that proteins do in the cell membranes. Now, in anatomy, we learned some structural things. For example, I made a big point in anatomy to talk about with you the structural phenomenon of the protein. So we talked about cell junctions. Can you name some of those? Proteins that build junctions. How cells interact with each other, cell-to-cell interaction. Gap junctions, which build protein holes between cells, like in the heart. Tight junctions, yeah, the zippers between the squamous epithelial cells and the brain. Desmosomes, those anchor-looking phenomenon with bars between the cells holding them together. They're found in the skin, helping to give strength to the epidermis. Okay, so joining cells together, we looked at those. I didn't make a big deal about it, and we won't talk much more about it in this class, but the shapes of cells are described because of proteins. Now, in this case, they're not the proteins in the membrane per se. They're the protein networks that are found in the cytoplasm of the cells. So most of these guys are built from tubulin, a protein that builds the network. So when I look at a nerve cell under a microscope, I can tell that's a nerve cell. It has a skeleton, and that skeleton is built out of protein. Okay, so all of that just to emphasize, there's your desmosomes, your tight junctions, the things that we talked about in the fall. Now, we're going to shift gears at this point from the anatomical kinds of phenomena that we find surrounding proteins to talking more about how the proteins actually function. What are the functional proteins? And we're going to learn in this class functional proteins from two basic perspectives. We'll expand it. Two basic perspectives. One through the lens of the nervous system, and the other through the lens of the endocrine system. So I've got these here listed for you. I'll come back to this. One through the lens of the nervous system, one through the lens of the endocrine system. In fact, if you have very clear in your mind how the nervous system functions at a molecular level and how the endocrine system functions at a molecular level, there's really nowhere you can't go. That was a double negative, I should say it this way. If you learn those two, you really can talk about any physiology you want to talk about. These are the two major systems in the body that run everything. And they have to work with each other as well. Now, in order to talk about how these two systems work at this level, which is what we're going to do in this class, this is what we would say at the molecular level, then you're going to have to understand what the proteins do that are in the cell membranes. Now, for both nervous and endocrine, and this will apply to others. You'll see it all over the urinary system as well. We'll get there next. If you're going to talk about the functions of the proteins, you're really going to talk about, for the nervous system, these proteins doing one of two things. They either serve as channels for ions, or they serve as pumps for ions. So both of these are illustrated here. Actually, these are the channels. So you can see that there's these red dots here passing through this protein from the outside to the inside. You can see here some square dots here. This is an endocrine example shown here. But the reason I show you these pictures is because one of the core principles in physiology with functional proteins is diffusion. Every student needs to come to neurophysiology, and by extension, every other chapter, with this very clear in their minds, simple diffusion. Now, we learned simple diffusion in freshman biology is something going from higher to lower, from an area of higher to an area of lower concentration, and that's true. But for us, the big ticket items passing through these channel proteins are charged ions. There's a reason why you have to have proteins to do this function. Lipids, the core of the membrane, the cell membrane, is nonpolar. Long chains of fatty acids. And so they don't interact well with things that are charged. So if you take, for example, if this red dot here is a charged sodium ion, it carries a +1 charge, +1 valence, and you bring that red ion into the proximity of this cell membrane, like this one is in close proximity to the cell membrane, it's not going to go through that cell. It will not interact with the nonpolar regions of the membrane. It may interact with the phosphate heads, the polar regions on the inside or the outside of the membrane, but it won't pass through here. It's charged things and uncharged things do not interact well with each other. This is a principle that we'll use over and over and over again. And because the membrane is nonpolar and these charged ions are polar, they carry charges, you cannot get them to move or to interact with each other, to move across them. In other words, I can't get this sodium ion to move across this membrane because of the chemical nature of it. Therefore, you have to have proteins that serve as channels for those ions. So as we begin neurophysiology, as we start our discussion of neurophysiology, this is going to be the first major point, identifying the important ions for making the nervous system work and the proteins that allow those ions to move across cell membranes. So, as it turns out, the first two we're going to talk about will never go away. You'll learn them today in class. We'll still be talking about them in May. They'll never go away. Sodium and potassium. Na and K, sodium and potassium, both of them carrying a +1 valence. They must, in order for anything to work, I'm going to say this again in just a second, I'll put a picture on there to make me say it more than once. In order for anything to work in your body, sodium is going to have to go across cell membranes, and so is potassium. You have to be able to get those ions to move inside and outside, and the way that happens is through protein channels. Okay, now, in addition to serving as protein channels, proteins also serve, so these are going to be protein channels as well that we'll do in the endocrine system. In addition to that, proteins, this one is out of your textbook, proteins serve as pumps. So the first principle of physiology for functional proteins is diffusion. Ions move across cell membranes through ion channels that are proteins. The second principle of physiology for proteins is active transport. In diffusion, things move from higher to lower concentrations. In active transport, it goes the other way. Things move against their diffusion gradient in active transport. And this picture walks you through the most important protein in human physiology. Now, that's a pretty dramatic statement. But you'd be hard-pressed to find anybody that knows anything about molecular biology or human physiology or neuroscience or endocrine science who would disagree with that. Kidney function, liver function, blood pressure regulation, heart function. Find a spot you want to talk about. This is where the conversation begins. The function of the sodium-potassium pump. And so this picture right here illustrates this, and we're going to walk through this as we get to it. I just want to introduce it to you today. The sodium-potassium ATPase pump. Now, I just said another word. In human physiology, you can have proteins that serve as ion channels that don't require energy. The channel is just open. The channel is, because of its chemical nature, selective. So it may only let sodium go through it. It's a channel there and it likes sodium, and sodium will go through it, and you don't have to do anything. It doesn't need any energy to do that. It goes from a higher to lower concentration by simple diffusion. Active transport is different. With active transport, you have to have energy. The illustration that Mary uses on the modules is getting the water to either go uphill or downhill. So she's got a little man with a little bucket. She stands on top, dudes on top of the hill, and pours the water out, and it diffuses. It runs downhill. No energy is required. But if the water's in a pool at the bottom of the hill, you have two options. You can either run a pipe from a pump to the top of the hill and run the pump, which provides the energy to push the water uphill, or the dude's got to get his bucket, fill it up with water, and carry it up there. Either way, it's going to require energy, and cellular energy is provided in the terminal phosphate bond on ATP, adenosine triphosphate. Now, very early in the textbook, the process of ATP generation is talked about. I will do that with you. We will go through the outline of glycolysis, the Krebs cycle, and oxidative phosphorylation when we get to muscles. So I will teach you, before this class is over, the mechanism by which phosphate groups are added to ADP and energy production. You're going to learn that in this class, but you're going to learn it in the context of muscle contraction. But I would argue that the number one reason in your body for energy is to run this pump. If you want to talk about muscle physiology, we're going to do that, right? How do muscles work? How do muscles contract? Guess what I'm going to start with? Sodium-potassium pump. You have to establish grades for sodium and potassium. If I'm going to talk about how the heart muscle contracts, this is where I'm going to start. If I'm going to talk about how your kidneys work, that's coming in this test section. How do your kidneys get water to go where it's supposed to go and electrolytes to go where they're supposed to go? This is where we're going to start. How is sugar balance done in the body? How does the kidney regulate sugar? This is where we're going to start. The function of this protein, the sodium-potassium ATPase pump. And as you can see here, the sodium-potassium pump transports with each phosphate group that it burns. ATP here is converted to ADP, adenosine tri to adenosine di. A phosphate group here is lost. The energy from the breaking of that terminal phosphate bond drives the function of this pump. It will take 3 sodium ions out of the cytoplasm and put them in the extracellular fluid. And without burning another ATP, it will take 2 potassium ions out of the extracellular space, put them in the cytoplasm. 3 to 2. 3 to 2. Every time the pump does what it does, it transports sodium by active transport out of the cell, and potassium into the cell. Now, having learned that, tell me, if I were to measure the concentrations of sodium and potassium around heart muscle, or around skeletal muscle, or around a neuron, or around an epithelial cell, anywhere in your body, what would I find right now in terms of concentrations?
There's more sodium.
There's more sodium
There's
outside the cell,
yeah.
and there's more potassium inside the cells. This is human physiology phenomenon. You don't work without it. Your brain won't work, your muscles won't work, nothing works unless sodium is more concentrated outside the cell, potassium is inside. Now, we're going to make the biggest case about this in nerves and muscles because they make action potentials. We're going to do that Thursday. That's going to be the biggest thing for us. How does the signal get from the precentral gyrus to the lower motor neuron in the ventral horn, then sending a signal by way of the musculocutaneous nerve to the bicep, right? How does that happen? And the answer is going to be the movement of sodium and potassium ions. It's electricity, really. In its basic function, we think of the nerves as a wire. But the wire in that wall right there is moving electrons. Your wires don't move electrons. They move current. Sodium and potassium, sodium and potassium, sodium and potassium, sodium and potassium. That is the wiring of the human body. And Hodgkin and Huxley ran a series of experiments back in the 1950s where this was discovered. You're going to get to reproduce them in the laboratory by simulation. Reproduce what they did. When you get to the end of it, you're going to go, that is so easy. That's so simple. The principles are so simple. Why did it take the human race to the 1950s to figure this out? Well, I don't know the answer to that question, but it is fascinating to me, this process. And it's very elegant. It's not hard to learn. It's fundamental. And so I like to start the class with it. The function of the sodium-potassium pump. You can't do anything without it. The most important protein. Okay, now I just quoted this. I got ahead of myself a little bit. You guys learned the brachial plexus, and you learned what you learned in anatomy were the connections between the brain and the spinal cord, and then from the spinal cord, by way of the nerves, the named nerves that we got, to the muscles. And you learned these pathways, as I just spouted out. Precentral gyrus, down the lower motor neurons, and the cord, out through the nerves to the muscles. And then I showed you a picture, when we were in anatomy, a picture of the nerve communicating with the muscle. Now, we didn't talk about the details of it, but we will do that in this class. After we've learned the nerve function, then we'll go to the muscles and learn the muscle function. And so I just wanted to I want you guys to think about this in context. In anatomy, you learn the pathway. In physiology, you're going to learn the mechanism by which that signal got from one place to the next. Now, the first piece of that is going to be the electricity, the current. We'll talk about that. The second piece of that is going to be, what about places where there are gaps? So the muscle is one of those places. So the musculocutaneous nerve, we learned, controls the bicep. But the musculocutaneous nerve does not touch the bicep, even though it looks like it does. In fact, there's a gap between the end of the motor neuron and the muscle cell membrane. So we're going to have to do something in that gap. The gaps, or the synapses, are dependent on the function of a whole group of molecules for which you're going to learn about 20 of them. There's probably 60 or so now known in science. You're going to learn several of them. The first 4 are the big ones, and you're going to learn those well. These are called neurotransmitters, getting the signal from one neuron to the next. Here comes the current, blow the chemicals out. The chemicals now go over here, and they start the current again. Neurophysiology, the transmission of information in the human body. Okay, now, the endocrine system is going to come later, but since it's so important, I'd like to introduce it here. I taught you about it near the end of the semester in anatomy, and we talked about the different ways cells make things. We said you can make them in an exocrine fashion, and you got ducts. Or you make them in endocrine fashion, you don't have ducts. So I like to bring it up at the beginning of class here. The nervous and endocrine system are really going to serve as our core for physiology. Nervous is coming first. Then we'll do hormones in the test 2 section. We're not going to do hormones in the first test section. But we have to keep tying these together. Physiology is one of those places where you're really not going to be able to learn something about one thing and then not have it somewhere else. This is one of those things where you just have to take it all with you as you learn pieces and parts. So the endocrine system, as I pointed out to you, is made up of a bunch of glands that release things into the cardiovascular system. And you learn, for anatomy, the connection between the hypothalamus and the pituitary. I did that on purpose with you so that you can start thinking about endocrine function. Now, from what you've heard from me today, if I'm going to talk about endocrine function, how it works, what class of organic macromolecules are going to have to be at the center of that? There's no way around this. Proteins are going to have to be at the center of this. So in the nervous system, our major focus in the nervous system is going to be the proteins that let sodium and potassium go across the membranes, either by diffusion or active transport. When we get to the endocrine system, you will have already been introduced to it in part in the nervous system, but we'll really start hammering away at it. In the endocrine system, our focus is going to become the proteins that serve as receptors for chemicals, hormones made in the body. So in the nervous system, the introduction is going to be like this. When the action potential gets to the end of the nerve and it wants to make the muscle contract, the way it does that is it releases a chemical, a neurotransmitter, that will then bind to the other membrane and make action potentials on that one. So that's going to be our nervous system. When we get to the endocrine system, these glands are going to make molecules. For example, we'll just keep it close to the adrenal gland. The adrenal glands, the adrenal medulla, produces a hormone called epinephrine. We know it as adrenaline. And it releases that adrenaline into our system. It's part of the sympathetic nervous system, which you know a lot about already. And that adrenaline's going all over you. It's jacking your heart rate up. It's changing your blood pressure. It's reorganizing the way the liver releases sugar, doing a lot to you for your survival. How? That's the physiology question. How does it do that? Well, in order for adrenaline to do its thing, it's going to have to bind to receptors in the cell membrane. And guess what those things are? Organic macromolecule? Those are proteins. So in the nervous system, the proteins are going to be ion channels. They're going to be pumps. When we get to the endocrine system, they're going to serve as receptors. Proteins will be receptors. They have ways of recognizing. These proteins recognize these hormones. And when the hormones bind to these proteins, it sets in motion a whole cascade of events. This is physiology. What are those events? Why is that applicable? Why would you want to know how epinephrine binds to its receptor and what happens after that? This is pharmacology in today's world. The pharmacologists don't give drugs and hope they do something. They know the molecular and biochemical mechanisms. You know, if I know that epinephrine binding to the beta-1 adrenergic receptor activates a cyclic AMP-dependent kinase pathway, well, I'm going to design a drug that acts on the pieces and parts inside that cell. One of the best illustrations for this is all the drugs available for acid production now. So we'll do this in the digestive system. So the hormones come in, they bind to the gastric cells, the epithelial cells, and they cause them to ultimately make acid. Well, these are going to be the parietal cells. And now we know all the molecular mechanisms. And guess what all the molecules are that begin and run that process? They're proteins. The receptors for histamine, and gastrin, and acetylcholine, those are all proteins. They're ion channels. They move hydrogen ions and chloride ions. And so there's a whole litany of drugs now available that go after that mechanism from all different perspectives. Once you've learned that in this class, you will never look at the back of a Nexium or Zantac box the same again. You're going to want to know, well, is this an antiport thing? Or is this blocking histamine binding? Or is this acting on the acetylcholine pathway? And all these drugs go after it a different way. You know how they did that? By learning the physiology behind it. One more illustration here, and then I'm going to do feedback mechanisms with you. There's a local physician here in town, Dr. Jonathan Forrester. He's a bit of a nut, just so you know. I love him. He's a dear, fabulous nut. And we go to church together. And I love listening to him because he's crazy. But he's crazy in the most beautiful way. I don't have a better way to say it. He's involved with this group down at LSU New Orleans, has been for the last 4 or 5 years, working on this treatment of cancer process. Now, it's moved along quite nicely. And I can tell you the whole story. It's a very fascinating story. But we're not talking about today. What I want you to hear is the fundamentals of physiology that are driving this. And so what was discovered in the lab down at the med school in New Orleans, a couple of guys down there discovered that sodium-potassium pumps and channels for sodium ions are not just in nerves and muscles. They're also in epithelial cells. Now, a large percentage of the cancers that kill human beings are epithelial cancers. And what they discovered was that epithelial cancer cells overproduce the channels that let sodium go into those cells by orders of magnitude. Now, in the nervous system, if you stimulate a neuron, you cause some sodium channels that were closed to open. You'll learn them later. They're called voltage-gated sodium channels. They discovered that there were orders of magnitude more of these guys. So they administered a drug to these epithelial cancers. That blocked the function of the sodium-potassium pumps in transporting the sodium out of the cell. They would stimulate the cell with current, literally put a live wire in the petri dish. That would open up the voltage-gated sodium channels and flood the cell with sodium. The pumps are blocked, so the cell cannot get the sodium back out again. The water in the extracellular space follows the sodium. And guess what happens to the cell? It bursts. Now, this is also a very common way for your body to get rid of cancer cells. I'll teach you this mechanism when we do immunology later in the semester. So, by just knowing the fundamentals of physiology, these people stand on the cusp of making a major contribution to the treatment of cancer. There's a group right now, a collaborative effort between the physics department and the vet school at LSU Baton Rouge right now, building a body device that they can put on dogs to give stimulation to them, to try to mimic what they've done in vitro with these cancer cells for stage 4 cancer cells for epithelial stuff. Now, it is possible, and I think this is what Dr. Forrester is banking on, it is possible that they will be able to develop a device that will do just that. Now, they're going to have to deal with the kidneys, because once you kill a bunch of cells like that, the kidneys have got to get rid of them, and you're going to clog things up if you do that. And so there's some issues they're going to have to deal with, and they're going to kill some dogs doing it. But if they develop a body stimulation thing and an appropriate drug to block the sodium-potassium pumps, it is possible that in the next decade, there will be single day to 2 week experiences for people that will get rid of stage 4 epithelial cancers. Man, that would be really cool. And it started with what you've learned today. The function of the sodium-potassium pump and the voltage-gated sodium channels let the sodium pass through them. Okay, so nowhere we go, we'll be able to get away from it. And I wanted to emphasize that with the endocrine system as well. Now, I taught you this in the fall. We'll come back to it for the control of hormones. And then we'll talk about the proteins that they bind to, and I'll give you that. And then the final thing for this introduction, before I can start with neurophys, is to talk about feedback mechanisms. So here are the principles. Let's do them now together. The principles to open physiology with. The first major principle I wanted to get across today for you was, if we're going to explain how it works, you got to talk about proteins. The second major principle was, okay, well, what proteins are you talking about? And I'm talking about sodium-potassium pumps and channels. These are the proteins we're going to emphasize in the nervous system. And when we start talking about hormones, we're going to talk about receptors. So this is the way I want you thinking about proteins right now. I want you thinking about proteins as pumps for ions, channels for ions, and receptors for molecules. Those three will get us done for human physiology. Now, in terms of general physiological function, we really need to think about feedback mechanisms. And I threw this slide up in the fall as well, but I want you to keep this in the back of your mind as we talk about various physiological things during this semester. And both the nervous and endocrine illustrate these. I'll point them out. We can really think about feedback mechanisms in the human body mostly as negative feedback systems. Most of the stuff that works in you and me is done this way. Our bodies are very intent on maintaining a balance of things. Your body temperature doesn't need to get too high. Your blood sugar levels don't need to get too high. There's probably 10,000 of those things. We just started listing them off, all the things that have to be regulated, especially if you go to the molecular level. Thousands and thousands of things that your body regulates. That process of regulation is something that every intro A&P course needs to define. It's called homeostasis. And I'm hoping you heard that word as a freshman student. Homeostasis means to keep things in balance. Keep it in balance. And the way humans keep things in balance is with an ebb and flow. By balance, you do not mean it stays constant. By balance, you mean it stays within a range. So, how many times have you taken your body temperature and it was exactly 37 degrees Celsius or 98.6? I can remember, years ago, first learning that and then thinking to myself, I don't think I've ever taken my temperature and it was 98.6. It's because our temperature does this. We keep it in balance by way of a negative feedback system. That system, incidentally, you will learn. What is the name of the nucleus in the hypothalamus that regulates body temperature? Does that sound familiar when I say that? The preoptic nucleus is the body temperature regulator. So we'll want to return to that, yeah? How does it regulate temperature? What are some things that your body does to regulate temperature? Let's say you're too hot. What happens? You sweat. Why?
Because when the sweat evaporates, it cools your skin.
Yeah, because somehow the autonomic nervous system, in its connection with the preoptic nucleus, knows about the latent heat of vaporization. Right, physics students? How much energy is lost whenever a liquid molecule becomes a gas molecule? I'll learn this in physics.
Depends on the big or the specific heat of the whatever it is.
Yes, but average. Okay. Okay, so that just don't get caught. That's not a fair question. So let's don't get caught up in that. The point is to say your body's doing things to regulate the temperature. Your body does things to regulate sugar levels. Can you name the number one hormone that reduces body sugar levels? Insulin.
What do you mean?
You talking about the insulin receptors? Is that what you were saying?
That's not right.
Insulin is the number one hormone. Okay, so everybody knows that one. What is the number one hormone that elevates sugar levels when your body sugars are low? Now, what is it that everybody would know about insulin and nobody comes to me knowing about glucagon? Why is that? Yeah, because everybody knows someone or has heard the story about a diabetic. And diabetics either they don't make insulin at all, type 1s, or, so that makes up about 10% or less of the total diabetic population, or their receptors for insulin, which are what kind of molecules? Proteins, don't work right. So they have insulin, but the insulin won't bind the receptor. Or if it binds the receptor, it doesn't function like the kinase like it's supposed to. So negative feedback systems, that's number one. Now, there are other feedback mechanisms that are important for us. For example, there are some things in the human body, that operate on a positive feedback phenomenon. That is, once the process has begun, it's hard to get it stopped. But childbirth is a great example of that. So the production of oxytocin initiates that process probably by binding to the smooth muscle in the cervix And the pressure then begins a positive feedback cycle that results in childbirth. And think of another way. Now, I didn't tell you this a minute ago, but this one right here, this process right here, by which the ion balances are established and the voltage-gated channels open to produce an action potential, that is actually a positive feedback mechanism. It is going to become for us a phenomenon that we call the all or none phenomenon for nerve and muscle function. Look, once you get it to a certain point, you can't stop it. The nerve's going to work, the muscle's going to contract, the heart's going to beat, the gland's going to release. This is how we work, positive feedback system. There are a couple other big ones that have come up this semester. Another huge one in this class is blood clotting pathway. So normally when you cut yourself, there's this molecule that comes out of the connective tissues into the surrounding liquid around the blood vessel called tissue thromboplastin. It's a blood clotting factor. Now once that stuff gets into the system, you're not going to be able to stop the clot formation. Because each of the subsequent clotting factors is initiated by the one before it. And once the first one is made, it's coming. A clot is coming. So clotting is another good one. So there are others in the immune system as well that are important. But that one's going to come up in this class, and I'll teach you the clotting cascade. So I wanted to mention it today. Okay, so there are negative feedbacks and positive feedbacks, and that's what your book says. Now, in addition, let's move ahead. So I'll just talk through this with you. Positive feedback. Let's finish with this for today. I'm going to start this. I'm going to give you the categories we're going to work with this picture, and then on Wednesday I'm going to start going through the categories. So there is another important feedback mechanism that the book just doesn't. No book talks about it, and we're going to talk about it on more than one occasion in here. And this is a phenomenon in your body called timing. As it turns out, the human body has the ability to tell time. I could take any of you and put you in an underground bunker where you had no access to the outside world, no radio, no TV, nobody's telling you what time it is, you don't know when the sun's coming up, when the sun's going down, and I could put a camera in there and watch your behavior. And in the first day, these things start to arise very clearly in watching people. They wake up and they go to sleep very predictably. For humans, they will wake up and go to sleep, they get active and non-active on a cycle of about 25 hours. There are variations, but it's real close. Human beings have clocks that run slow. Your brain thinks the world revolves, the earth is turning at a pace a little bit slower than it is. It's a very interesting phenomenon because most bird and mammals and everything down to blue-green algae that's been studied has the ability to do this. Fruit flies, there's loads of information on Drosophila, even the genetics are worked out on those. They have the ability to tell time without timing cues. Now why? Why would living things have the ability to tell time? We're not going to try to answer that question, but it's an interesting one. Ponder that in your free time. Second question is this. We know that they can tell time. Why is it that they don't tell time on a 24-hour cycle? If the world that we grew up in has always run at 24 hours, why is it that every living thing that we look at tells time non-24 hours? That's an interesting question. Not for today. Now here's the third one. everything has a clock that we study. Everything that has a clock has a clock that runs, not coordinated with the regular timing of the planet. Then, third question is it important that our clocks run in sync with the world that we live in? And that's the point for the feedback mechanism. You know the answer to that question, don't you? Is it important that your clock runs in sync with the world you live in? How would you prove that?
I get fired if it doesn't.
What's that? Okay, fall asleep in class. All right. Something more fun, some more fundamental things. How do you know this is important? How do you know this is important feedback? How do you know this is important for human physiology?
Your circadian rhythm and how your light helps you stay awake and puts you to sleep.
Why? Why would you need that? Why can't you? Why can't your body rhythms be out of sync with the sun and the day and the night? Can you think of things that? Can you think of illustrations in the real world that you know of that would illustrate you can't operate like that?
Sometimes when the time change and stuff, it's really hard to keep up. So I think it's kind of linked to that because
How would you really focus in on that question? Time change. Yeah. Get in an airplane and fly to Europe. Yes? You're 7 or 8 hours off. How are you going to feel the next day? Yeah, weird and funky. How about this one? The incidence of depression is related to latitude.
Yeah, because people that live in really, really far to the top, they don't see as much sun, so it makes them more depressed.
Well, in the summertime, they see a lot more sun.
Right. But typically it's in the winter.
Winter's longer. Yeah, so we know that timing is related. You know you're going to be sleepy if your timing is off. There's a whole list. You can type this up online. Incidents caused by timing problems, clock problems, major disasters caused by this. I would argue that most major disasters are related somehow to this feedback mechanism. You have to have, your clock has to be synced to the world you live in. So I'm going to pay attention to it. Now, as it turns out, you had a good anatomy class. So you already know, you guys already know how your clock is synced to the time zone that you live in. How do you, your clock is running at 25 hours. That is not going to work. Your clock needs to run at 24 hours, which means it has to be reset every day. How do you set your clock? You learned the pathway, didn't you? Light by way of the retinal hypothalamic tract into the hypothalamus. And what is that structure? The suprachiasmatic nucleus. Exactly. You've got to send the timing information into the clock in the hypothalamus. And then the clock has to distribute that timing information downstream. As it turns out, almost everything in your body ebbs and flows, not only in a homeostatic way, but also in a daily way. Things rise and fall on a 24-hour cycle. And they don't all rise and fall together. Sometimes things rise during the day, sometimes things rise during the night. Sometimes things rise at the midday. And so what has to happen is somehow your clock mechanism in your body has got to orchestrate a plethora of 24-hour cycles so that they're all kept in balance. Which is why I bring it up during the first class of human physiology. I would argue with anybody that your ability to tell time is an important physiological, basic, fundamental physiological process. Negative feedback, positive feedback, and timing. Last thing, categories for learning neurophysiology. We're going to build categories for learning physiology by learning about 3 groups of proteins. So I want to finish class with this and start here on Wednesday. 3 groups of proteins that will help us think about neurophysiology. The first group I've already introduced to you. These are the group of proteins that serve as ion channels, passive ion channels. They're proteins in the membrane, and they're selective now. They don't let anything go through them. They let particular ions go through. And the 2 we're going to focus on are sodium and potassium. We're going to call them passive channels. Proteins in the membranes. Now, you're going to have to know where in the membranes they are, these ion channels are, and when they're working, and how they establish charges. We'll do all that on Wednesday. The second group of proteins are also ion channels, but they're normally closed. We'll call them voltage-gated channels. That is, you have to do something to the voltage, the charge of the cell, in order for them to open. And again, you'll have sodium voltage-gated and potassium voltage-gated channels. The third group of channels that we'll talk about on Wednesday are the ones that I will call the chemically gated channels. Now, there are several varieties of these, but the fundamental variety of chemically gated channel is the channel that opens when a chemical binds to it. So the first one we'll learn is the one for acetylcholine for muscle contraction. When acetylcholine binds to this channel, it opens and allows sodium and potassium to pass through it. Passive, voltage-gated, and chemically gated. Those will be the categories that we will learn the functional pieces of neurophysiology with starting on Wednesday.